Now, we have learnt a lot, right? Literally since morning, gut, and we have been talking about genes, we have been talking about methylation, we came to stem cells, peptides, metabolomics, what all and what not, right? What if we make it simple now? Simple and nice, easy to understand how to read the genes, the DNA. Can we have a genetic testing like that? And that too, which is actionable. Come on, we don't always want rocket science, isn't it? Come on, all that data, too much complexity we don't want, right? We want something actionable, and that's why the next speaker, who's also lovingly called Dr. T, by the way, created something. When he saw in his practice that people do not understand all this, they want to discover things. And yes, just like in the previous session by Dr. Itakshi, she was talking about how much the client of hers was confused with gymming and weight loss. And that was the exact reason that he was reversing. He was, just going around in circles. Now, if you can discover such a genetic testing, which can help you exactly get to know about your nutrition, your lifestyle, your exercise and protocols, etc., why not that, right? So are you ready for the next session, which will be taken by Dr. Thangadurai, who literally researched for this and found out that you can have some actionable testing. And let me also tell you, this is also available in his exhibit outside, okay? And you will get to know about that. So please welcome Dr. Thangadurai to the stage, a skilled functional medicine doctor who has definitely helped the mission of helping practitioners to help them use genetic testing in the real world. Let me help you. You can. Thank you. Thank you, Dr. Thangadurai.
Wellness evening, everyone. I'm not hearing anything. Yeah. You got the energy still. Yeah. Yes. Yeah, it's okay. So when I talk about genetic testing, what comes to your mind? You may think of any indications. Particularly, you will think of infertility, right? And some birth defects or early mortality running in the families. Other than that, we don't even think of genetic testing, right? The science is growing, but the medicine is not growing. There is a knowledge gap between the science and medicine. And this is what we are doing. We are translating the science into clinical practice. You'll know how. So this is what called the central dogma of life, right? The flow of information is always unilateral. That is what we think. And from DNA to RNA and RNA to proteins. Right? So we think that DNA is what everything is happening around. And we forgot that the genes themselves cannot turn off or on. Isn't it? So what happens here? It's the environmental signals that the cell receives through its thousands of receptors and perceive the signal and act accordingly. The genes are the blueprint. Okay? Am I clear? So, the Human Genome Project is the pinnacle of modern medicine. And I will give you some valuable gifts for the keen listeners and the smart people. I'm going to ask you one question. How many DNA letters are there in human genome? Anyone? I mean totally. In the entire genome, how many DNA letters are there? Anyone? A quick guess, 6 billion letters. 6 billion letters repeated with only 4 alphabets. That's what God has coded us, right? Okay, this is a cosmic joke. With Human Genome Project, what we were thinking was, if you are able to look at the genes, we will find out the solution for each and every conditions, right? There are more than 1 lakh of proteins in our human body, so the scientists expected more than 1 lakh gene before starting the Human Genome Project. But what happened? There were only less than 25,000 genes, it's decreasing day by day, isn't it? Everything we evolved through epigenetics. So most often, only few disorders and diseases are caused by genes. This is called disease-linked genes. But other than this, most of the conditions are caused by the gene-environment interaction through epigenetics. Could you see here? This is not a one-way, this is two-way, right? And it's the environment that decides everything. Okay. There is one thing that is more important than the environment. What is that? How you perceive things. Can we do one simple thing? Can I? Shall we? Yeah. Could you all please close your eyes for a while? Please. Please, please. Yeah. Now just think of honeybee. How many of you thought of honey? Please open your eyes. And how many of you were scared of honeybees? Could you see that? That is the placebo and the nocebo effect. It's more than the environment, it's your perception. And the perception depends totally on the mind. So train your mind. Do you think epigenetic decides everything? Everyone? Okay. What else? Environment is epigenetics? Yes? Anything else? So we assume that environmental conditions, food and lifestyle decides everything, isn't it? But we assumed that there is no defect in your DNA, right? But what if your blueprint is defective? You have to know the code first, then only you can act accordingly. Fortunately, the food, the environment, the life you lead can compensate and even complement for the genetic imperfections. That is what God has gifted us. So it's always not the nature versus nurture, but always a gene-environmental interaction, right? So this is how we feel when we look at the genetic panel, isn't it? Just like opening the hood of your car. And we don't understand much. Why? Because it is not studied well. And it is not a diagnostic test actually, right? It's a test for knowing the disease susceptibility. And how many of you have done the genetic testing for your clinical practice? Please raise your hands. Only few? Okay. So the results, when you look at the genetic test results, is it empowering you or disempowering you? When you're doing it for yourself. Disempowering for many of the fellows. Because we will be looking the genetic panel in terms of green, blue, or else like red, right? So it disempowers us because nothing is given with respect to the epigenetics, controlling the gene. Only it is given that whether the gene is defective or not, or else nothing is given, like polygenic risk score. You all know about it, right? What is polygenic risk score? They will give in terms of like percentiles or in terms of odds ratio, something like 2.1 or 1.7, something like that. We can't make anything out of it, isn't it? Okay. So we also don't know about the enzymes coded by the genes, whether it is fast or slow. There is no information about that. And also the epigenetic regulators, whether there are some inhibitors and enhancers with respect to the gene that is not being given there. And there is no actionable insights. We need to change this. If we change this, we can integrate genetic testing into clinical practice so that we'll be having a very good results. Probably you'll be having 70 to 80% success rate with respect to all the conditions. Yes? And if you integrate genetic testing, it will be close to 90 or more than that. I would say genetic tests are not useless, but useless. Because when we look at the genetic testing, the geneticists or the genetic counselors will be like focusing only on the disease-linked genes. We are not looking at the disease susceptibility, right? Those genes are so important, that is what is causing most of the chronic conditions, isn't it? Yeah. How many of you are using methylation in your clinical practice? Raise your hands, please. Great. And how many of you are giving B12 shots for your patients? Please raise your hands. Only this much. And how many of you are giving chelation? Very good. And how many of you are using the ordinary foundational supplements? These things works with epigenetics, with methylation. Okay. So another quick question for you. How many percentage of your DNA is derived from viruses? Yes? Very great. I would like to give you a superfood for her. It's actually 5 to 8%. Well done. And then, how to interpret the genetic test report? How the ideal test report should be like? You have to look at the teamwork. The genes don't function individually or singly, right? It works actually in a community. But each has been given a different roles, and each gene does a lot of roles. It's not only one role that has been given to the gene. And we have to look at the pathways, right? We are not looking at the biological or biochemistry pathways nowadays. We are looking only at the genes, and if at all something is there, only supplementation is being recommended in most of the genetic testing, unfortunately. So what to look at the genes? We have to look at the substrate and the product, and we also look at the epigenetic factors which induces or inhibits them, and also the cofactors. For example, in this case, we're all aware about the MTHFR mutation, right? Isn't it? What happens with that? 3677T, anyone? See, what has happened is simply a substitution of C cytosine to thymine has reduced the efficiency of the enzyme down to 30% and 70% for heterozygous variant and homozygous variant. Okay? So what has happened here is, simply because of the nucleotide substitution, there is a translation in the proteins, which means like the alanine is been changed to valine, right? So there are 656 amino acids in the MTHFR gene, okay? That is the protein. But a single change in the amino acid can reduce the efficiency of the enzyme by 70%. You all know about this? Could you see the alanine and valine here? Yes? So we have to look at the epigenetic factors as well. Can you see this? That is the homocysteine being converted to methionine through MTR, right? So we have to look at the epigenetic factors here, that is the heavy metals inhibiting those things. And another quick question is, what is the source of acetaldehyde other than alcohol in your body? What is the source of acetaldehyde other than alcohol? Anyone? Sugar? No, I just want to be in the clinical scenario. No? Anyone else? What kind of infections? Who told that? Yeah, great. I want to give that thing, man. Another energy drink. Yeah. That is SIFO, right? Small intestinal fungal overgrowth. Yeah, great. And we have to look at the bigger picture. We should not focus only on the genes, but also the upstream and the downstream pathways as well. This is more important. Okay? So the core concept is, we have to look not at the diseases, not at the genes, but the abnormal biochemical pathways. Because we don't transmit the diseases as such, but we transmit the abnormal biochemical pathways through your genes. So what is the stepwise actionable steps? We have to look up the teamwork. And we have to remove the clogs, and we need to look at the epigenetic regulators here. And the effect of SNPs, whether it slows down the enzymes or fosters up the enzymes, right? And we can replete the cofactors or deficiency, but this one should be the last step. Unfortunately, we are only looking at the food or the supplementation, right? This is not the way it works. Yeah. So even when someone is having, that is the G6 vitamin D protein polymorphisms, what happens is you may have very low vitamin D levels, but there is a normal calcium metabolism happening. In this case, you may not have to boost up your vitamin D supplementation. What happens when someone has FADS polymorphism? Anyone? Will he be able to convert ALA to EPA and DHA? No. And unfortunately, FADS polymorphism is associated with increased inflammatory markers because of the metabolites of arachidonic acid. And how many of you know that only 10% of cancers are inheritable? Rest are all sporadic. So what is the cause of sporadic cancers? Anyone? Anyone? Anything else? Yeah, defective mitochondria is fine. Anything else? You just look at there. When there is a, do you think the tumor suppressor gene or the proto-oncogene causes cancer? No, it is not. Okay? Unless it is mutated. Unless it is mutated, no tumor suppressor gene or a proto-oncogene causes cancer. Are you all excited to know what causes cancer? Yes? So we look at the hormonal pathways. Okay? How many of you are wearing xenoestrogens here? Throw your cosmetics. How many of you are actually using the organic cosmetics here? No one? Great. Keep doing it. And here you can clearly see most of us are having the same exposure, right? Then why someone is acquiring the cancer at an early age? Because of the inherent capacity to metabolize the estrogens. Here you could clearly see it can be metabolized through CYP enzymes into 2-hydroxy and 4-hydroxy estrogens. 4-hydroxy is having more affinity towards the DNA, and when it is metabolized to semiquinones and quinones, it can form DNA adducts. And the COMT gene and the NQOY actually comes into rescue, and these can actually bring it back towards the hydroxy estrogens or methyl estrogens, which means when someone is suffering from, when someone is having a gene mutation in the CYP1, A1 or CYP1B1, which is fast mutation, and when it is getting clogged here, that is when you are not having adequate enzymatic activity happening at COMT and NQO1, what happens is there is accumulation of semiquinones and quinones, right? Then it causes mutation. Is it clear? So this could be explained for most of the estrogen dominance you see, and also for the cancers, hormone-sensitive cancers. As I have told you that glutathione here is an antioxidant reducing the toxicity of the semiquinones and quinones. And you could also see here that the TNF-alpha induces CYP enzymes and inhibits COMT, which means it promotes mutation. Okay, this is some kind of a scoring. You can look into it. And do you like this? Pathways? Yes or no? Definitely no, right? Now? Is it clear now? See, while looking at these pathways, do remember one thing. There are some sources, and see, for example, there is some precursor for dopamine, and there are some receptors actually, and there is a reuptake, and also there is metabolism. In this way, when we look at each and everything, it is easy to sort it out. Same thing with serotonin pathways. So most of the mood disorders, psychiatric conditions can be clearly explained by this. Unfortunately, we are not running these panels for most of the chronic, mood disorders and psychiatric conditions. And there is a lot of controversy about glutathione, right? You all agree about that? So what is happening is, a lot of glutathione is good for you or bad for you? Good for you? Definitely? Okay. Could you see here clearly that glutathione is inhibiting its own enzyme? Here? Are you seeing here? GCLC? Why it is happening? See, when you're giving a lot of glutathione, it will be converted to lot of cysteine and glutamate. The cysteine is actually converted to lot of, what is this? RNS. Anyone? Nitrogen species, right? So this is what is causing a lot of damage to the mitochondria. And also your body. This is what we have to understand. Lot of glutathione is not good for you. And how many of you are running the allergic panel? Most of you, I think. Isn't it? Are you getting good results with that? It never happens. Because when you are having multiple sensitivities, you should not look at the allergic panel, but rather you have to look at the histamine pathways. Okay? So simply we have to look at the source of histamine here and how it is metabolized, and it acts via the receptors here, and it can be deactivated in this way, and it can be excreted. So if you divide this one, it's so easy, isn't it? So what is the drug that inhibits DAO? Anyone? Quick question. What is the drug that inhibits DAO? Please, yes please. It's the most commonly used medicine. Anyone, anything else? I'm sorry, it is metformin. Okay. How many of you are prescribing folic acid to your clients in the pre-conception period and also for the antenatal care? Most of you, right? Why we should not recommend folic acid here? Anyone? See here, the folic acid itself inhibits DHFR and also the MTHFR. Why should we recommend this unmethylated? This is synthetic one. The folic acid is synthetic one. It should never be recommended. The one take-home message you should all carry is you should never recommend folic acid for antenatal care. Okay? What happens when you recommend folic acid? There is something called unmetabolized folic acid, right? What it causes, it increases the risk of running the cancers. Okay? Yeah. This is again methylation pathway. So why we are so much focused on methylation? Because most of the SAMe are used for the synthesis of phosphatidylcholine and creatine through PEMT and GAMT. This is so much importance. What is the most important gene for pregnancy? Anyone? Why is phosphatidylcholine important? It's an integral part of the cell membrane. So if you want to grow the fetus well, we need a lot of things. That is here it has been induced, right? Yeah. And I don't want to give we have to only look at the longevity. I want to look at only the longevity. So I will focus only on longevity. It never happens, okay? I already told you genes functions as a community, right? So you have to know that the longevity genes are nothing but the antioxidant gene and the genes which are involved in the cellular stress and defenses and which checks inflammation and which are having very good DNA repair. For example, the PONR peroxonase will actually alleviate the fatty liver and also atherosclerotic lesions here when it is like fast. Okay. So there are two pillars of longevity. We all know about that. That is the positive lifespan gene and the negative lifespan gene. The positive lifespan genes are involved in DNA repair. There should be strong DNA repair, strong RNA transport, and there should not be much of a high energy metabolism and not much of high inflammation. You all know, excited about this? Homeostasis? Can anyone tell what is homeostasis? Yes, a good stress that builds resilience in you, isn't it? Again, why we should not recommend too much of fasting or too much of exercise? We need to find a sweet spot, right? Even with exercise and fasting, we should not overdo it. And what is the number one priority for longevity? Anyone? Fasting? Anything else in common? Exercise, right? See, you had to get out of your comfort zone. You had to simulate some adversity to your genes. Your genes should be perceiving the adversity. Otherwise, you will die earlier. Okay? Can we all hold our breath for a while, for a minute or two? Is it possible? Do it. You are stimulating your genes. That will improve your survival every day.
10 minutes to go.
So we all know about the mTOR and the insulin pathways, signaling pathways and sirtuins, right? And this is what xenohomeostasis is. This is so much important. And there is one molecule that is a xenohormetin that is used everywhere. Anyone else? Anyone want to know? Answer this question. What is a xenohormetin that most of us know? Anyone? It's a red wine. But we can't have xenohormetins like drinking red wine because we need 600 glasses of red wine to get the same effect. So we don't undergo stress, but also the plants also undergo stress. And what are xenohormetins? The plant undergoes stress, and you can actually get all these beneficial markers, beneficial polyphenols, in a way like you had undergone a similar stress. So the resveratrol is what I have told about the xenohormetins, and it works via sirtuins. Anyone know about matcha? Why it is different from green tea? Anyone? No, it is shadow grown for the last 20 to 40 days to make it more stressful so that it can elaborate a lot of polyphenols. That is the way it works with olive oil also. Okay. So the NAD levels usually drop when we age. So we need to boost up the NAD levels through NMN more than NR. And here you could clearly see this resveratrol acts as a stack. What is stack? Anyone? Sirtuin activating compound. Okay? And we need fuel, right? What is sirtuins? Anyone? What are sirtuins? Yes? Okay. What are they? Yeah. NAD dependent? What is that? Histone? Deacetylesters. So we need to have sirtuins. We need to have the fuel first, and then we have to click on the accelerator pedal, isn't it? So the key to longevity is again your low insulin levels every time and high insulin sensitivity, and we had to give moderate calorie restrictions in the environment of micronutrient excellence. Again, telomere, we all know about that, and all these things, happiness, gratitude, positive outlook, self-love, in service, having a purpose, this will actually induce your telomerase and make you live longer. So don't retire early, not even in your 60s or 70s, right? You all stay young, be active at your age of 100. You all have to celebrate your centenarian birthday, right? Okay, we have introduced a panel called DNA Flex in which we look at all these things elaborately, and will be giving a lot of epigenetic control points. And I just want to know, and I just want to give some invaluable supplements here. So, when there is multiple allergies or food allergies, what is the pathway you have to look into? Oh my God, everyone is. I can't give it for everyone. Okay, fine. So, what is the length of your DNA? Anyone? What is the length of your DNA? Anyone? It's very simple. Yeah, that is the total one. 2 meters. Excellent. I want to give this young man. Yeah, natural immunomodulator. So this is like 6 feet. Okay? And we stretch our DNA through. And then when we stretch our DNA, We are having 37 trillion cells. We can go to the sun back and forth 600 times. Okay? So you have to know your code first. You can upgrade your clinical practice, and it could be like the precision medicine, the future of healthcare. And thank you all for your future patient listening. And I just want to ask one more question. How many percentage are we different genetically? 0.01. Who told that? Yeah, excellent. Thank you all.
Thank you. Thank you so much, Dr. Thangadurai. And thank you so much for such an interesting presentation. No doubt about it.